A very good morning again to all of you. Good morning everyone. Today the topic that we are going to do is a continuation of what we did yesterday, because the principle about which we are going to talk, that is Archimedes principle, for that we will be needing a slight information about what we saw yesterday, that is about pressure, right. Yesterday's class I told you, someone of you asked about buoyancy, that why are we not including buoyant force instead of pressure forces. So I said that buoyancy is a consequence of pressure difference in fluids, right. So yesterday I told you that how the liquid pressure changes with height in an incompressible liquid. So when you go down the liquid, so the pressure increases by rho gh, right, and any point which is exposed to atmosphere, its pressure is PATM. If there is no atmosphere or vacuum is there, that means the pressure will be 0. So we will quickly take one or two questions on that, and then we will come to the concept of buoyancy. So suppose we have a vessel, right, and here we have two liquids. They are immiscible, they cannot be mixed with each other. So this is the second liquid. So we have got two liquids over here. Now the liquid which you are seeing on the top, this liquid has a density rho 1, this liquid has density rho 2. Now this height is h1 and this height is h2. So if we are asked what is the pressure at the base, atmospheric pressure is given as P0. We are taking a second case where the same vessel again is drawn. This time what we are doing is, we are having a lid, not lead. So this is a lid, or you can say it is a piston, and its cross section area is A and its mass is m, and here again we have the two liquids. This is rho 1, this is rho 2. Height again is h2 and h1. Outside pressure PATM we are taking as P0. So again the case is, what is the pressure at the base? So now if you see the first case, very simply we can solve such questions. How we can solve? Just in one line you can write, if I select this point as A, this point as B and this point as C. So here when we solve such questions, just remember these all questions are to be solved in one line. How? When they are asking pressure at base, so I will write pressure at base, you can compare 2 points of the same liquid. B and C are of the same liquid, so pressure at base is nothing but the pressure at C. Now if you see the C and B points, they are of the same liquid, and going by the formula that we learned yesterday, so we can say C will be at higher pressure than B. So I will write PC is nothing but PB plus rho GH. Is it clear? PB plus rho GH. Now which density will I use? Since it is of the second liquid, I will write rho 2 and H2. That's it. Now if I see PB, PB is the pressure of point B. I can compare it with A. If I compare it with A, you can see PB is lower than PA, so it will be of more pressure. So PB can be written as PA plus rho GH again. Which liquid is common for both A and B? It is the liquid 1. So I will write rho 1 GH1. And what about PA? A is in contact with atmosphere, yesterday I told. So PA is nothing but PATM. So in short, when we write the pressure at the base, we will simply in one line we will write, just go on coming down. Here pressure is P naught, here it will be P naught plus rho GH. Again plus rho GH. So what will I write? P base is at the top pressure is PATM, which is P naught. Then come down in first liquid rho 1 GH1. Then again you are coming down in second liquid rho 2 GH2. That's it. This is the pressure. Is it clear? So when you come down, just you remember the case of equilibrium. So you have to keep on increasing the pressure as you go down. So as you are coming down, you are seeing that the pressure is keeping on increasing by what value? Rho GH. Clear to everyone? Okay. Now the same thing if I apply here, the case is slightly different because here it was open to atmosphere and here we have kept a piston whose area is A and it is also having mass. So what will happen? There will be an extra pressure apart from atmospheric pressure. There is an extra pressure which is pushing the liquid down, and that is because of the weight of the piston. Because of the weight of the piston will try to come down, and in doing so it is pushing both the liquids. But since the system is in equilibrium, so net force should be balanced. Okay, so by that pressure keeps on increasing down. If I write the pressure over here in the second example, what will I write? Pressure at the base. Just see in one line again, how will I write? If I see the point A, so come down, first you will write pressure at base is equals to, start from the point A of the liquid. Now when you come down, pressure is increasing by how much? Rho GH. Which liquid? First liquid, rho 1 GH1. From point A you reach the point B and again you are coming to point C. Now again you are coming down. So plus rho 2 GH2. Is it clear? So the pressure at base will be PA plus rho 1 GH1 plus rho 2 GH2. Now the problem here is, in the previous case PA was P naught. Why? Because it was open to atmosphere. But is the point A over here open to atmosphere? The answer is no. So what is the pressure at point A? If you see the pressure at point A, you will say it is equals to, first of all there is atmospheric pressure which is pushing it down, and there is pressure due to weight also. Now what is the pressure due to weight? Pressure due to weight is this piston's force is mg and divided by area. Those who did not get it, I am showing it again. If this is the piston, from outside atmosphere will exert a force, atmospheric pressure is P naught, so it is pushing it down. And if you see the weight of the piston, this is also acting down, mg, and I have taken this point as A, so pressure at the point A multiplied by area of cross section will push the piston up. So piston is being pushed up and two forces are coming down. So when you balance the forces, so I will say net force on the piston is 0. That means upward force is balancing the downward force. So from here only we get PA is equals to P naught plus mg by A. So those who had confusion in this, I have written over here clearly what is the reason, but while you solve question, you won't be having so much time to waste that every time you will write these all things. So simple here, pressure at the base is equals to P naught plus mg by A. Start from here, the pressure is P naught. Now you came down, so you came down, pressure increased by how much? Mg by A. Mg is the weight divided by area, force by area is pressure. So mg by A. Again you come down, rho 1 GH1. Again you come down, rho 2 GH2. So this is the entire pressure getting added and the final pressure at the point C will be this. Is it clear to everyone? Please check it once. Okay. So we will now move on to what we call as Archimedes principle. So you saw here the cases. Now let's move and see Archimedes principle. So Archimedes, he introduced the concept of buoyancy. So when we talk of buoyancy, what do we mean by buoyancy? So when an object is put inside a liquid or inside a fluid, apart from experiencing its own weight, there will be one force which will try to push it up. There will be one force which will try to push it up. That upward force which is exerted by the liquid, we are talking of the net upward force exerted by the liquid. Now many students, or I think presently I can surely say that 100% of the students would reply incorrectly if I ask the definition of buoyancy, right? Because why I am saying so? Because what happens that it is generally misinterpreted. People think that buoyancy means upward force. Buoyancy does not mean upward force or upthrust by the liquid. It is the net upthrust, because when you are inside a liquid, yesterday I told that the liquid above the object will try to push it down and liquid below the object will try to push it up. So there is a downward force also due to pressure and there is upward force also due to pressure. So when we find the net upward force, why I am saying net force is upwards, because we saw yesterday that whenever we take inside a liquid 2 points, here the pressure at the base pressure is more than at the top. So what happens that the force by the liquid from the bottom and force by the liquid from the top, which force will be more? This one will be more. So it will try to push it up. The net upward force is called as buoyancy. The net upward force, it is not just the force. I always disagree with anyone who tells that the upward force is called buoyancy. Wrong. The net, I always use the word net, means resultant. So the resultant upward force exerted by the liquid or fluid on an object is known as buoyant force. So Archimedes, he gave the principle about this. What he said that when any object is completely, again I will try to remove a few things which have been wrongly interpreted by most of the students till they come to say 11th standard. So they take the thing wrongly even in Archimedes principle. Okay, let me clear that. When any object is completely or partially inside a fluid. Now if an object is completely or partially inside the fluid, before writing further, let me ask you, will there be buoyant force or not? If an object is completely or partially inside a fluid, will there be a buoyant force or not? Let me hear first, then I will reply. It is not necessary, third time I told. Now listen why. Suppose you would have seen a piece of paper. Is it heavier or lighter than water? A piece of paper, is it heavier or lighter than water? If you just keep it on the top of water, or you just crumble and put it on water, it is lighter, yes, you know. So buoyant force will push it up and its weight will pull it down. But what happens that buoyant force is more, so it floats. So it goes up and then finally buoyant force balances the weight and it floats. That is the different thing. Now you would have seen sometimes that you can try this in your kitchen sink or your wash basin anywhere. You will sometimes see that in the wash basin, if this is the wash basin and this is the wash basin hole, this is the wash basin hole. Sometimes you will see a small piece of paper gets, keep a piece of paper over here. Put your hand on it, keep a piece of paper, now fill the wash basin with water. What will you see? Will the paper come up or it will stick there? Will the piece of paper come up or will it stick there? You would have seen even in kitchen many a times some things get clogged in the wash basin or in the kitchen sink. You will see that it will stick. Is it so or not? The answer is no. Why is buoyant force not acting? Because below the paper there is no water. Below the paper there is no water. There is water only from above. So what will the water do from above? So suppose if I draw this again and you are seeing that this paper got stuck here. Now you fill the sink with water. Is there any water below? Here you can see this is the pipe. Now what has happened? Above it there is water, but below there is no water. So is there any upward force? Answer is no. Now again I ask you, is it completely inside water or not? Yes, it is inside. But is there any buoyancy? No. There is only a down thrust. There will be only a down thrust. So what does that mean? Does buoyancy necessarily act on an object which is completely or partially inside a liquid? No. It will act only if there is some liquid placed below it. There is some liquid placed below it. Got my point? Now think, if suppose I lift the paper with my hands slightly, if I apply a force and I pull it slightly from above the hole, I remove it from the above the hole, will it stay there or now will it go up? If I take this piece of paper and remove it from the hole slightly and leave it, will it go up or not? Yes. Why? Because now water has come below it. Now water has come below it and the pressure force due to water below it, it will push it upwards. Is it clear now? Yes. So that means for buoyancy, when an object is completely or partially immersed in a fluid and fluid is present below the object also, then it experiences a net upthrust. It experiences a net upthrust which we call as B, which is known as buoyant force. This net upward force is known as buoyant force. So the object should have some liquid below it also, so it will experience a net upthrust, buoyant force, and this B, and due to it the object will experience a loss in weight because its original weight mg is acting downwards and there is a net upward force which is called the buoyant force. I am again saying net means resultant upward force by the liquid. So due to these two forces, the effective weight which will be measured will come out less than the true weight. So that is called as buoyant force. So whenever we have some object placed inside a liquid completely or partially and liquid is present below it, so it will exert a force, the liquid will exert a force upwards. Clear? So here the expression for buoyant force is equal to loss in weight of the object inside the fluid. So the loss in weight of the object inside the fluid is buoyant force. Now, what is the reason? I told reason of buoyancy is simply the difference in pressure at two different points in a fluid. So due to difference in pressure, what will happen, that the net force which is acting upwards, that we call as the buoyant force. and we have an expression for buoyant force. Buoyant force we say is equal to, as I said, loss in weight of object inside the fluid. So we call also it as V in rho L into G. Buoyant force is V in rho L into G. Now what is meant by that? See clearly. V in over here, what I tried or mean to say is volume of object inside the liquid, if we are talking about a liquid only. Rho L is density of the liquid. So buoyant force is equals to V in rho L into G, okay, where V in is the volume of object that is lying inside the liquid. Might be possible in some case only some volume is lying inside, not the complete one. In some case the entire volume can lie inside. So whatever be the case, buoyant force is volume lying inside into density of liquid into G. So basically buoyant force is due to pressure difference. So you have two choices. When you are writing the Newton's second law or first law, whichever Newton's law you are seeing, when you are showing the diagram and all the forces acting, if you are showing pressure force P into area, then don't show buoyant force, because difference in pressure forces will only be equal to buoyant force. So either you show the pressure difference force or you show the buoyant force. I'll tell you by example, but this thing should be clear that whenever it is placed completely or partially inside a liquid, the object will experience a net upward force, which is called as the buoyant force, provided some liquid is present below it, and the buoyant force is equal to the loss in weight of the object inside the fluid, and its value numerically is V in into rho L into G. I will show it why it is so. Okay, by two examples you will understand why we are taking V in, volume of the object lying inside. Okay, so let's see. Now I am taking two cases. Suppose I have this object, this is a solid object, and say I have tied it with a string, right? Now this is partially inside a liquid, and let's take this height as small h, total height as h. Clear? Okay. Now atmospheric pressure is P naught. This area I am taking as A, and density of the liquid is rho L. This is the density of liquid. Clear? Now let's see. If I draw the diagram over here, below the liquid there will be an upward force, let's call it as Fu, upward force. Can I say this is, suppose I take this point as 1 and let's take this point as 2. So can I say Fu, force at this point, will be pressure at this point, that is P1 multiplied by the area. Will I write so? I hope you agree to this. Okay. Now you can see here pressure is atmospheric pressure. Here, because it is open to air, this is atmospheric pressure. So what will be the pressure at point 1? How much below is it from the top? I will say here pressure is P naught. So can I say P1 will be P naught plus rho G small h? Agree to this? If here the pressure is P naught, can I say here the pressure is P naught plus rho G h? Yes. So I will write here P naught plus rho G small h into A. This is upward. Now if you see from the top, here there is a down thrust due to atmosphere. Now here it is open to atmosphere, it is not in liquid. So what is the pressure here? P naught. and what it is exerting downward force? P naught A. Is it clear? Now next thing, we will have this solid, it will have, suppose its mass is m. So you will have its weight downwards, mg. Is any other force missing in this? Please tell me. Is any other force missing in this? I have shown these three forces acting on this object. Is any other force missing? Please let me know first. Think, you will come to know whether a force is missing or not. Saying normal force. How normal force? There is a tension. It is not left freely. Can you see, here is a string. Here we have a string. So there will be a tension. So there is an upward force also by the string, which is tension in the string. These are the forces. Clear? Okay. Now what I am doing is, just see this diagram. I am redrawing and say, what shall I, if I write this is T and there is one more force which is mg, one more force mg. Now the net force by the liquid in upward direction, we call that force as buoyancy. Net force by the liquid in the upward direction is called buoyancy. And I told buoyancy is due to pressure difference, force due to pressure difference. Can I say? So either you draw the diagram like this or like this, both are same. But when you are drawing buoyant force, don't show pressure, and when you are showing pressure, don't show buoyant force. Now, if I compare these two, what is in place of what force did I write buoyant force? Can I say this buoyant force is actually, this buoyant force I have written in place of Fu minus Fd. Fu is pushing it up, Fd is pushing it down. So what is the buoyant force? Net upthrust. Net upthrust is Fu minus Fd. So if I subtract Fu minus Fd, tell me what are you going to write for this? Fu minus Fd, what are you going to write? Fu minus Fd, will something cancel? You can see P naught A, P naught A, cancel. P naught A cancels. What will be left? Rho G H A. Is this the value? Is this the value or not? Rho G H A. Correct? Now, if I write this, tell me, is the complete volume of the object inside the liquid or it is partially inside? Yes, it is partially inside. Can you see what is the volume inside? Volume inside the liquid is, can I say, area of cross section into what is the height inside, h. Can I write this or not? Everybody agrees to this? Let me know fast. Can you say that V inside is AH? Yes. So can I substitute AH as V inside? So did I get V inside? Rho was rho of liquid into G. Did I get a formula for buoyant force? Check here. So can you see that the same formula in the previous page I have written, B is equals to V inside into rho L into G. I am again using the same only. So buoyant force is actually due to pressure difference in the liquid. If pressure is changing, because of that the net upward force, not the upward. If I say, is this buoyant force? Fu is not buoyant force. Buoyant force is Fu minus Fd. Atmospheric pressure is exerting a force downwards P naught A and liquid is exerting a force upwards which is P1A. The difference in these two, which is pushing it upwards, that is known as buoyant force. And for buoyant force to act, what is the condition required? What is the condition required? It is required that there should be liquid present below the object and it should push it up. Is it clear to everyone? So has buoyant force anything to do with atmospheric pressure? Reply me fast. You are going to get a very important concept from now on. Has buoyant force anything to do with atmospheric pressure? Yes or no? Suppose I change the atmospheric pressure. Will the formula of buoyant force change? Yes or no? The answer is no. Why? Because you are seeing that P naught A cancels. Means buoyant force is independent of atmospheric pressure. It depends only on pressure difference, not on pressure. It is a consequence of pressure difference in liquid. It is not due to atmospheric pressure. Buoyant force is due to change in pressure, or it is a consequence of pressure difference. So it has got nothing to do with atmospheric pressure. Even if you take this object in a liquid and you take it in vacuum, still there also the buoyant force exerted by the liquid on the object will be V inside into rho L into G. Is it clear to everyone? So this is a very important point, and Tikshana, she was repeatedly writing that can buoyant force be negative? So please let me answer this thing. You this is self-evident thing, and you should know this thing, that which point will have more pressure, bottom or top? When you go inside a liquid, does pressure increase or decrease? When you go down, pressure will increase. Yes. So pressure will increase. So the force which will act on the base, will it be directed upwards or downwards? That will be Fu. It will be acting upwards, and the force on the top will be acting downwards. So Tikshana again reply, which force will be more, Fu or Fd? Will Fu be more or will Fd be more? So obviously Fu will be more. So upward force is always means more than the downward force whenever you have a situation like this. So that means what? Can buoyant force be negative? If buoyant force is Fu minus Fd, and this formula is valid only when liquid is present below the object. If there is no liquid, there won't be any buoyant force acting. So that clearly explains that buoyant force has to be positive only, and its direction is always where? Upwards. Understood? So we will stop the discussion over here because your next class is now going to start. I hope few things I made clear to all of you, so that when you will study the chapter fluid mechanics again, you won't have difficulty in understanding buoyant force. And what all things I told, kindly remember this thing. Okay, so that will be all for today. It was pleasure discussing these all things with you. Thank you.
Hello, good morning students. So in last class what we have discussed about types of isomers, structural isomer, that is chain isomerism and position isomerism. So today we are going to discuss next important part, that is functional isomerism. So that is most important part. So write down number 3 is functional isomers or functional isomerism. Now in functional isomerism there is no use of any parent chain or position. Important point is two compounds having same molecular formula but having different functional group are called as functional isomers. So what is important point here? Functional group will be different. So what if we write statement, what we write? Two or more compounds having same molecular formula, having same molecular formula but having different functional group, having different functional group are called as functional isomers, called as functional isomers. So now we have to remember that which pair of compounds can form functional isomers. So that is very important. If a given molecular formula is given, we have to think which of the following type of functional groups can form which type of isomer? Functional isomers. So let us make pairs. So first A pair is, or A part is saturated alcohol and saturated ether. Now what is meaning of saturated? It means there is no double bond or triple bond present. What is present? Single bonds are present. Alcohol means which functional group? OH, and ether means ROR. So with same molecular formula, both structures are possible, saturated alcohol as well as saturated ether. So let us take one example. If we write molecular formula C2H6O, this is a molecular formula. Now I can draw two possible structure for this. One is saturated alcohol and one saturated ether. So if I draw structure A, CH3, CH2, OH. Now if you count how many carbon atoms are present, 2, hydrogen 3 plus 2, 5, 5 plus 1, 6 and 1 oxygen. So molecular formula is C2H6O. And what is structure? CH3, CH2, OH. So it means what we have written? Saturated alcohol. And next if I write CH3, OCH3. So what we have written? Saturated ether. Now both compounds have same molecular formula but functional group is different. One is alcohol and one is ether. So what is the relationship between A and B? These compounds are functional isomers of each other. So they are functional isomers. So compound having saturated alcohol, it have also one isomer which is saturated ether. So these two class of compound, first example is for which isomer? Functional isomers. Any doubt in this? Saturated alcohol, saturated ether? Let us move to next pair of functional group. Next pair of functional group is B part. If we take unsaturated alcohol, unsaturated ether, next is cyclic alcohol, cyclic ether, aldehyde and ketone. Now for same molecular formula, these all structures are possible. What are possible? Unsaturated alcohol, unsaturated ether, cyclic alcohol, cyclic ether, aldehyde and ketone. Let us take one molecular formula. I am taking molecular formula is C3H6O. What is molecular formula? C3H6O. 3 carbon atoms, 6 hydrogen, 1 oxygen. And we have to draw all these possible structures. So let us first start unsaturated alcohol. Unsaturated means which contain double bond. So first structure I am writing CH2. We'll place one double bond here. Double bond CH, CH2, OH. So if you check how many carbon atoms are present? 1, 2, 3. How many hydrogen? 3 plus 2, 4, 5 and 6 and 1 oxygen. So CH2 double bond CH double bond is present. So it is unsaturated. And which functional group is present? OH. So unsaturated alcohol. Next we have to write unsaturated ether. So if we write unsaturated ether, CH2 double bond CH, OCH3. So this is ROR ether. And on one side if you check what is present? Double bond. So it is unsaturated and it is an example of unsaturated ether. Next we have to draw cyclic alcohol. It means ring is present and which group is present? OH group. So how many carbon atom? 3 carbon atoms. So if we write, so cyclic alcohol. Next what we have to write? Cyclic ether. If we write cyclic ether, so 4-membered ring, out of which 3 are carbon, 1 is oxygen. So ROR. So this is cyclic ether. Next is aldehyde. 3 carbon atom aldehyde. If we take CH3, CH2, CHO. And next is 3 carbon atom ketone CH3, C double bond O, CH3. So with same molecular formula, how many different functional groups are possible? One functional group is unsaturated alcohol. Next functional group is unsaturated ether. Next is cyclic alcohol. Next is cyclic ether. After that aldehyde and last is ketone. Now these are not total number of structural isomer. Just we have drawn which of the following are possible for this. We can draw different isomer for unsaturated alcohol. We can different for ether, cyclic ether. But this is only possible cases of functional groups. So what we have to remember here, that these are not total number of structural isomers. Not total number of structural isomers. So this is not point we have to remember. Sometimes whenever you see this question, how many isomers are possible for C3H6O, you will mark answer is 6. Answer is not 6. When we discuss questions on total isomers, then we discuss that for alcohol 1 more is possible, for ether 1 more is possible, then total will be 8. So that we will write down. But first important point are, these are not total number of structural isomer. What is there? Only 6 functional groups are written. Clear? So this is second B part, pair of functional group. Now if we move to next C part, now C part is primary amine, secondary amine and tertiary amine. I hope you have studied this in nomenclature, in starting of nomenclature, that what is degree of amine, what is degree of amide, for degree of alcohol. So if I write again what is meaning of degree of amine, it is equal to number of carbon atoms directly attached to nitrogen. So if nitrogen is directly attached to 1 carbon atom, then it is primary. If it is attached to 2 carbon atom, secondary. If it is attached to 3 carbon atom, then it is tertiary. So if we take example, first I am writing CH3. First I will write molecular formula. For which molecular formula we have to write? C3H9N. But we have written C3H9N. So if we start for primary amine, first structure I am writing CH3, CH2, CH2, NH2. Now if you check here, how many carbon atom are present? 3. Hydrogen, 3 plus 2, 5, 5 plus 2, 7, 7 plus 2, 9, and how many nitrogen? Only 1 nitrogen is present. So which type of amine it is? Primary amine. Because if you check, nitrogen is attached with 2 hydrogen and 1 carbon. So if it is attached with 1 carbon, so what is degree of amine? 1. So it is primary amine, that is 1 degree amine. Next, if I draw next structure B part, I will write CH3, CH2, NH, CH3. Now if you count how many total carbon? 3 carbon. How many hydrogen? 3 plus 2, 5, 5 plus 1, 6, 6 plus 3, 9. So same molecular formula C3H9N. But in this compound or structure, nitrogen is attached with 1 hydrogen and how many carbon? 2 carbon. So it is example of which amine? Secondary amine structure. Now if I write C part, CH3, nitrogen, CH3, CH3. So molecular formula is same, C3H9 nitrogen, and nitrogen is attached with how many carbon atom? 3 carbon atom. So this is tertiary amine. Now if you check, molecular formula is same, functional group is different. One is primary amine, one is secondary, one is tertiary. Now why they are considered as different functional groups? Because they show different chemical properties. And atom or group of atoms which determines the chemical property of any compound, that are called as functional group. So that's why they are considered as different functional group. Okay? So we can say this A, B and C structure are functional isomers of each other. So what point we have to remember? That primary, secondary and tertiary amine are functional isomers of each other. And these are examples we have written. One primary amine, one secondary, one tertiary, having same molecular formula. Next, move to next D part is amide functional group. Primary amide, secondary amide and tertiary amide. So first I will tell about that what is meaning of degree of amide and what is their representation. Representation for primary amide, representation for secondary amide, representation for tertiary amide. So what is degree of amide? So degree of amide is same as degree of amine. Number of carbon atoms directly attached to nitrogen. but in this amide carbonyl group is present. What is carbonyl? C double bond O is present. So if I write primary amide, so representation for primary amide is C double bond O NH2. Now if you check with nitrogen, 2 hydrogen are attached and 1 carbon is attached. So this is representation for which amide? Primary amide. Now on this side either alkyl group is present or hydrogen can also be present. So this is very important, that what is fixed part? C double bond O NH2, and on this side with C double bond O either any alkyl group is present or we can write hydrogen also. If we write representation for secondary amide, now for secondary amide what is representation? C double bond O NH. Now on this side what is present? Alkyl group, means CH3, C2H5, any carbon alkyl group is present. So with nitrogen, how many carbon are directly attached? 2 and 1 hydrogen. So it is which amide? Secondary. But with C double bond O, either R can be present or hydrogen can also be present. But here with NH we cannot write hydrogen. We have to write only R group. Because if you write here hydrogen, secondary amide is converted to which amide? Primary amide. Now if we write representation for tertiary amide, so it will be C double bond O N. On both sides valency is vacant. Now here what we have to write? Only R group. It can be same alkyl group or different. We are writing R1 or R2. What is R? R is alkyl group. You know, CH3 methyl, C2H5 methyl, that are called as alkyl group. Clear? R1, R2, and on this side what is present? Either R or it can be H. Now if you check with nitrogen, how many carbon are directly attached? 3 carbon are directly attached. So this is representation for which amide? Tertiary amide. So these representation are first important for structural isomerism also. So first remember this representation, then we take one example that how primary, secondary, tertiary amide can form structural isomer, that is functional isomer. So if we take example, we'll write CH3, CH2, C double bond O NH2. Now it is primary amide, secondary amide or tertiary amide? I have written first structure, you have to tell it is primary, secondary or tertiary. So it is which type of amide? Primary amide. Very good. And what is molecular formula for this? It is 1 degree amide. And if we write what is molecular formula for this, how many carbon atoms are present? 3 carbon atoms. How many hydrogen? 3 plus 2, 5 plus 2, 7. 1 nitrogen and 1 oxygen. Next, if we draw a structure CH3, C double bond O, NHCH3. Now this is structure for which amide? Secondary amide. And if we write molecular formula, same molecular formula, 3 carbon atoms, 7 hydrogen, nitrogen and oxygen. C3H7NO, nitrogen and oxygen, not only oxygen. Next, if I write hydrogen, C double bond O, nitrogen, CH3, CH3. Now this is example of which amide? Tertiary amide. And same molecular formula, it is C3H7NO. Now if you check, these 3 are compounds having same molecular formula but they are having different functional group. Primary amide, secondary amide and tertiary amide. So that's why they are considered as which type of isomers of each other? They are considered as functional isomers. So they are functional isomers of each other. Clear? So primary, secondary, tertiary amine as well as primary, secondary, tertiary amide are considered as which type of isomer? Functional isomers. Now let us move to next part, that is E part. So E part is carboxylic acid and ester. What is written? Carboxylic acid and ester. Now what is representation for carboxylic acid if we write? First I am writing representation for carboxylic acid. Which functional group is carboxylic acid? COH. So C double bond O, OH. Now on this side either alkyl group is present or hydrogen can also be present. So RCOOH, this functional group is called as which functional group? Carboxylic acid. Now what is ester? Now ester is R, C double bond O, OR dash. Here hydrogen can also be present. Now these two alkyl group can be same or it can be different. Now here with oxygen what is present? Alkyl group. Here with oxygen what is present? Hydrogen. So this is carboxylic acid and this is ester. So if we take example, let me draw first structure CH3, CH2, C double bond OH. Now which functional group it is? Carboxylic acid. And if we write molecular formula for this, what is molecular formula? How many carbon atom? 1, 2, 3. 3 carbon atoms are present. How many hydrogen are present? 3 plus 2, 5. 5 plus 1, 6. And how many oxygen are present? 2 oxygen. So molecular formula is C3H6O2 and we have drawn which structure? Carboxylic acid. Now this is compound A. If we write compound B, CH3, C double bond O, OCH3. Now this is ester. If you check R, C double bond O, OR. So which type it is? Ester functional group. Now how many carbon atoms? Same 3 carbon atom. How many hydrogen? 6. And how many oxygen are present? 2. So C3H6O2. So this is ester having same function molecular formula. Now if molecular formula is same but functional group is different, so which type of isomers they are? Functional isomers. Clear? So this is next type, carboxylic acid and ester. So these all pairs you have to remember. If you do not remember pairs, you cannot attempt these functional isomer questions. Now next pair is cyanide and isocyanide. Cyanide and isocyanide. Okay. Now which functional group is cyanide? Cyanide is also called as nitrile. Another cyanide is also called as nitrile. So which functional group? RCN. So this RCN is cyanide or nitrile. And what is isocyanide or isonitrile? It is RNC. So isocyanide group is nitrogen bonded NC and here carbon bonded CN. So if we take example, for example CH3CN and CH3NC. So what is the relationship between these two? These are functional isomers of each other. Clear? And one special case will come which are called as tautomers. Tautomers is example of special case of functional isomer. So HCN and HNC. So these are special case which are called as tautomers also. And tautomerism or tautomers is a special type of functional isomerism. So remember this example also, HCN and HNC, these also cyanide and isocyanide. So they are also which type of isomer? Functional isomer. But special name is they are tautomers of each other. Now if we move to next part, nitro and one is O-nitro. Or nitrito, sometimes it is written O-nitrito or O-nitro. So what is nitro? RNO2. And what is O-nitro? RONO. Only bonding is different. Here alkyl group is bonded with nitrogen. Here alkyl group is bonded with oxygen. So they are also considered as which type of isomer? Functional isomers. So these are different pairs of functional group which show functional isomers. Now I am repeating all these. You have to remember all these pairs. So first pair is saturated alcohol and saturated ether. So same molecular formula, these two structures are possible, saturated alcohol and saturated ether. Now next pair is unsaturated alcohol, unsaturated ether, cyclic alcohol, cyclic ether, aldehyde and ketone. So this is B part, which contains 6 functional group that we have to remember. Next pair of functional group is primary amine, secondary amine and tertiary amine. Next pair of functional group primary amide, secondary amide and tertiary amide. After that next pair is carboxylic acid and ester. After that cyanide and isocyanide, and one is this group nitro and O-nitro. So if we remember all these pair of functional group, then we are able to identify which pair of compounds are functional isomers of each other. Now there are some note point in this. Now degree of alcohol is also defined: primary, secondary, tertiary. What is difference between representation of nitro? This is difference. Here bonded with the nitrogen, here bonded with oxygen. So different type of bonds are present. Alkyl group is bonded with nitrogen, alkyl group is bonded with oxygen. So different type of bonds are present. Now degree of alcohol is also defined: primary alcohol, secondary alcohol, tertiary alcohol, but they are not considered as functional isomer because their chemical properties are similar. So first point what we have to remember, that primary, secondary and tertiary alcohol are not considered as functional isomers. So if I write this example, let us say CH3, CH2, CH2, OH. Which type of alcohol it is? Primary alcohol. And if I write CH3, CH, OH, CH3, which type of alcohol it is? Secondary alcohol. Now they are not functional isomers of each other. Now you have to tell what is the relationship between these two compounds, which type of isomers these are. I have written two structures, one is for primary alcohol, one is for secondary alcohol. You have to tell what is type of isomers these two compounds are. Which type of isomer they are? Position isomer. Length of parent chain is same. So they are position isomers of each other. They are not functional isomers, they are position isomers. Clear? What is reason? Because length of parent chain same, 3 carbon atom, 3 carbon atom. Here OH is present at first position, here OH is present at second position. So do not consider degree of alcohol as functional isomer because they have similar chemical properties. Now next, if we write number 2, if we take two compounds, benzene CH2OH and one is written OH, and here what is present? CH3. What is molecular formula for these two compounds? First you have to tell what is molecular formula. These two structures are written and you have to tell what is molecular formula. Not asking which type of isomer. First we have to check either they are isomers or not. So we have to check their molecular formula is same or different. So first you have to tell molecular formula. How many carbon atom, how many hydrogen. Now let us check how many carbon atoms are present. If we take 6 plus 7 carbon atoms, how many hydrogen are present? 1, 2, 3, 4, 4 plus 3, 7, 7 plus 1, 8. And what is present? Oxygen. Now this is C6H4 plus CH3 and plus OH. So it is C7H8O. And if you count here also you will get same. How many carbon? 1, 2, 3, 4, 5, 6, 7 carbon atom. How many hydrogen? 1, 2, 3, 4, 5, 6, 7, 8. So how many hydrogen are present? 8. So molecular formula is same for both. Now which type of isomers you think these are? Which type of isomers? Which type of isomers these are? Chain, position or functional? We have studied 3 type of isomerism. This is very important that these are functional isomers of each other. What is relation between them? These are functional isomers of each other. Why they are considered as functional isomers? Because if you check, this is benzene directly attached with OH. This is aromatic alcohol. Which type of alcohol it is? Aromatic. But if you check, this is not a aromatic alcohol. OH is not directly attached with benzene. So they will show different chemical properties. They will show different chemical properties. How they show different chemical properties? If you take simple ROH and if you add NaOH to it, there will be no reaction. It does not react with NaOH. But if you take phenol and add NaOH to it, then phenol will react with NaOH. And what is formed here? ONa plus H2. So alcohols both are same. One is aliphatic alcohol and one is aromatic alcohol. They are showing different chemical properties. That's why they are considered as different functional groups. So remember this, if one is aromatic alcohol, OH is directly attached with benzene. That type of alcohol are called as aromatic. Benzene is a aromatic compound. And if OH is not directly attached with benzene, that is not aromatic alcohol. So between these two, what is the relation? They are functional isomers if their molecular formula is same. Any doubt in this? So these 2 points we have to remember. So first 8 pairs for functional isomers we have to remember, and then these 2 note points we have to remember. So this is about functional isomerism. Now next is, if we move to 4th type of structural isomerism, that is metamerism. Okay. Now metamers are formed whenever there are different alkyl groups along the polyvalent functional group. Now what is meaning of polyvalent functional group? Polyvalent means having valency more than one. For example, if we take, let us take ether group, oxygen. On oxygen, how many alkyl groups can be attached? 2. So it is a bivalent. So whenever there is any polyvalent functional group, and on that polyvalent functional group, if different alkyl groups are present, then which type of isomerism arises? Metamerism. So what we write, this type of isomerism arises. Type of isomerism arises due to different alkyl groups across polyvalent functional group. Across polyvalent functional group. Okay. If we take example, let us take first example is CH3, CH2, CH2, oxygen, CH3. So which is functional group here? Ether, oxygen. It is a bivalent. 2 valencies are present here. On one valency, which group is present? Propyl. On second valency, what is present? Methyl. Now if we change this, on both sides, if we apply methyl group, CH2, CH3. So C2H5, oxygen, C2H5. Now on polyvalent functional group, only alkyl groups are changed, but their molecular formula is same. Now if we write molecular formula for these two, how many carbon atoms are present total? 4 carbon atom. How many hydrogen are present? 10. And 1 oxygen. So it is C4H10 and O. Now both have same molecular formula, but they have different alkyl group. Functional group is same, but alkyl groups are different across oxygen. So that's why they are called as metamers. What is the relation between these two compounds A and B? They are metamers of each other. So what is condition for metamerism? First we have to check there should be a polyvalent functional group. And on that polyvalent functional group, different alkyl groups should be present. Clear? So we'll write that what is list of functional group which are considered as polyvalent functional group. So first example is clear. Next example, if we write, first I am writing which are polyvalent functional groups. Polyvalent functional groups. So these all groups we have to remember. For example, what is written here? Which functional group? Ether. Next functional group, if we take carbonyl, C double bond O. Next functional group we are taking C double bond O NH. Which amide it is? Secondary amide. Next functional group is C double bond O N. Which amide it is? Tertiary. है ना. So they can form metamers into if these alkyl groups are changed. We are not writing secondary and tertiary. Between tertiary metamers are possible. Next, if we write NH2 valency vacant, so it is secondary amine. Nitrogen 3 valency vacant, tertiary amine. Next, if we take C double bond O, which functional group? Ester. So all these are polyvalent functional group, and across this, what is possible? Metamers is possible. No, secondary and tertiary amines are not metamers. In secondary amine, there are possible cases. For example, C3H7, NHCH3. This is secondary. C2H5, NHC2H5. This is also secondary. Both are secondary, and in secondary amine, their alkyl groups are different. I am not saying that secondary and tertiary amine are metamers. In secondary amine, if alkyl groups are different, then they are metamers of each other. Is that clear? Who is asking that doubt? That secondary and tertiary they are functional isomer. In secondary, we have to check that. If different are present, then what is possible? These are metamers. So these are some example we have written for amine. We can write for same ester, tertiary amine. Any doubt in this metamerism? So it is very simple. What we have to remember, these polyvalent functional group, if alkyl groups are changed, then they are called as which type of isomers? Metamers. So mainly 4 types of isomers we have studied, and these all are important. Ring chain isomerism and tautomerism. Tautomerism we'll discuss in later part when you come to offline classes. Then we study tautomerism. In next 2 classes, what we discuss, first is one part left, that is ring chain isomerism, and after that we'll do combined question on total number of structural isomer. So before coming to next class, revise all these type of structural isomers so that you will be comfortable in calculating total number of isomers. So students, thank you for today. We'll meet in next class.
Good morning, students. Okay. Yesterday we have discussed about cell cycle. Do you remember cell cycle? What are the 2 phases of this cell cycle? I'll proceed with the questions. Later we'll go to the actual topic. 2 phases. Names I want. Okay. Interphase and M phase. Very good. The 2 phases are interphase and M phase. M phase is also called as mitosis phase, mitosis or divisional phase. Yes, correct. In interphase, how many subphases are there? 3 are there. Very good. What are those 3 in the sequence? In the sequence, which is the first one, second one, third one? Sequence. Very good. G1, S, and followed by G2. Which is the lengthiest phase among these 3? Lengthiest, which will take more time among these 3. Correct, G1. This is the lengthiest phase. Lengthiest phase because it will take more time. Now, you know, among these 2, which takes more time? Interphase or M phase, which takes more time? First one or second one? It is interphase. Okay. Yesterday I told you human cell, to complete the cell cycle, it will take certain time period. How much time will be taken by the human cell for this cell cycle? How much time? 90 minutes, 24 minutes, 24 hours, 24 seconds. 24 what? 24 hours. In that, cell division proper will take how much time? Division. I am not asking interphase. Division will take how much time? M phase is called as divisional phase. 1 hour. Very good, very good. Cell division proper lasts for 1 hour. 23 hours is there for interphase and 1 hour is there for the M phase. Yeah, very good. How much time is required for the yeast to complete the cell cycle? How much time? 90 minutes. Good. If a cell stops division, it enters from G1 to, it enters to some phase. What is that phase? If the cell wants to stop the division, very good, G0, very good ma. So it enters from G1 to G0 phase. It is also called as quiescent stage. Very good. Quiescent stage. Okay. Now let us go to the actual topic of today, that is mitosis, which is nothing but M phase or divisional phase. So in the mitosis, one cell will be entering, and finally after the mitosis you will be getting two daughter cells. One cell will become two daughter cells. And this mitosis, it involves two phases or two stages are there in this. They are called as karyokinesis and cytokinesis. What do you mean by karyokinesis? Yesterday I told you. What do you mean by karyokinesis? Karyo means carry on, meaning division of nucleus. Yes, kinesis means division, carry on means nucleus, nuclear division or division of nucleus. Very good. Cytokinesis, correct, division of nucleus, correct. Nuclear division is karyokinesis. Cytoplasmic division is called as cytokinesis. Okay? Yeah, cytoplasmic division is called cytokinesis and karyokinesis is nuclear division. So these are the two stages of the mitosis. For the sake of convenience, this karyokinesis is divided into four stages, which are called as prophase, metaphase, anaphase and telophase. So karyokinesis is divided into four phases or four subphases. They are called as prophase, metaphase, anaphase and telophase. So these four will be there in the karyokinesis. Actually, the changes will not be happening like in one stage. We can't demarcate the stages. It is a continuous process, but for our convenience, we are categorizing them into prophase, metaphase, anaphase and telophase to study the main events. But it is a continuous process. So there are transitions between prophase, metaphase and anaphase and even anaphase to telophase. Transitions will be there in between the phases. We can't clear cut the demarcation of these phases. Just for the convenience sake, we are learning like this. Okay. There are transitions in between them. Now, as I told you, mitosis, one cell will enter. The cell which is entering is called as parent cell. And it will generally happen in the diploid cells. The parent cell is generally diploid cell. And after the mitosis, this cell will form two cells. These two are also diploid cells. And these two are called as daughter cells. So by mitosis, one parent cell will be forming two daughter cells. And these two daughter cells are identical to each other and they are identical to even the parent cell also. So whatever the point you take, okay, everything, chromosome number, chromosome size, genes, everything they'll be same. Cytoplasm, cell organelles content, everything will be same. So because of that, this mitosis, as it is producing the daughter cells which are similar in all the aspects, this mitosis is called as equational division. Equational division. Because it is producing the daughter cells which are identical to themselves and to the parent cells also. As it is producing identical daughter cells, it is called as equational division. And it involves two phases, that is karyokinesis and cytokinesis. Karyokinesis, where nuclear division will happen, and it has 4 phases: prophase, metaphase, anaphase and telophase. Okay, yeah, equational division, very good. Now let us see what is going to happen in these phases. What are the changes that will happen in the prophase? What are the changes of metaphase? What are the changes of ana and telophases? After this, the cell will enter into the second step, that is cytokinesis. Okay, understood everybody? Let us proceed to the division, how you are going to see that prophase. So all of you can see here clearly mitosis stages. The first one, it is not the stages in the mitosis, this is the part of cell cycle. So in the interphase, the chromosomes will be looking like this. Interphase, the chromosome will be looking like this, blue color and red color one. And you already learnt that in S phase, DNA will be replicated. DNA will be replicated. So here you have one more blue color strand and red color strand, but we can't see them clearly. Okay, after S phase you can see the 2 strands, but as they are not condensed, we can't see them clearly. Now from here onwards, it is the mitosis. First stage is the prophase, first one, second one, third one and fourth one. So in the first one, prophase, all of you can see here that each chromosome is having 2 chromatids. Each chromosome is having 2 chromatids, but we can't see them clearly. Okay. In the metaphase, you can see that chromosomes are attached to these small thread-like structures called as spindle fibers. In this picture, that's what you can see. Then this chromosome, this blue one chromosome is splitting into 2 parts, 1 and 2 chromatids. They are getting separated. Red one, here we have in the cell 2 chromosomes, one is red one is blue one. But here you see in the anaphase, they are becoming 4 chromosomes are there, blue one 2 and red ones 2, they are dividing. Then in telophase, the 2 nuclei, these 2 are called as nuclei. This blue one is coming here and red one is coming here. The same case even here. So out of the 2 chromosomes, after splitting, they are forming the 4 chromosomes, and among the 4, 2 is entering into one daughter nucleus and the remaining 2 are entering into the another daughter nucleus in the telophase. Let us see how this is going to happen. All of you can see here interphase. Interphase will be showing the chromatin reticulum. Chromatin. Okay, chromatin is uncondensed form of chromosomes. Uncondensed genetic material is called as chromatin. That will be there in the interphase because it is not dividing. It is taking rest. Then in the prophase, what is happening is condensation will begin. This chromatin is changed. Chromatin is thread-like and you can see that chromatin has been changed into chromosomes. Now these are called as chromosomes. All of you can see clearly here nuclear envelope is there, nuclear membrane or envelope. That nuclear envelope is ruptured here. You can see the gaps in between here, gaps are there. And then the centrioles which are duplicated in the interphase, they started moving in the cytoplasm towards the opposite pole. These are the main events of the prophase. Let us see the prophase events. Prophase, which is the longest phase. That means in karyokinesis it will take more time. Okay, longest phase. And the changes which will happen in this, the first change is chromatin condenses into chromosomes. The meaning is this type of chromatin will be turned into this type of chromosomes. They are short, rod shaped, reduced chromosomes will be there. You can imagine a long thread, that is the chromatin. After folding, it is called as condensation. You can imagine like folding only, that is called condensation. By removing all the water molecules from that structure, the chromatin will be condensed to form the rod shaped thick chromosomes. And in that chromosomes, you can see that left half is called chromatid and right half is called as chromatid, and they're actually connected by the centromere. You know that these two are chromatids and this is centromere. Okay. So, chromatin will be transformed into chromosomes. That is the first event. So, chromatin, which is elongated form or expanded form, will undergo condensation to form the thick rod shaped chromosomes, which are held together, that each chromosome will be having two chromatids which are held together by one centromere. This is the first change that is going to happen in the prophase. Next, the second change. After this, here what you have seen is nuclear envelope is disappearing. So, nuclear envelope, then nucleolus, Golgi, ER disappear. Okay. The cell organelles like Golgi, ER, Golgi apparatus, ER, nucleolus, which is the amembranous structure of the nucleus, nuclear envelope, which is having two membranes, outer nuclear membrane and inner nuclear membrane, all these four disappear. That is the next event. And in the animal cells particularly, only in the animal cells, you know, centrioles are features of animal cells. So, in the animal cells, in animal cells only, centrioles, okay, the duplicated centrioles particularly, centrioles got duplicated in S phase, no? That duplicated centrioles migrate to opposite poles, migrate to opposite poles. So, here you can see that two are there here. Here one pair is there, here one pair is there. They started moving towards the opposite pole. They are coming towards the down area. To the other pole they are coming. So, the three major events are there in the prophase. The first event, condensation of chromatin to chromosomes. The second event, only in animal cells, the duplicated centrioles migrate to opposite poles. It is there only in animal cells. But in all other cells, nuclear envelope, nucleolus, Golgi and ER disappears. Okay. So, three events are the major events of the prophase. Have all understood, ma? Yes, very good. So, three events will be happening, and in the same way, something like asters you will be seeing in the textbook when you refer that. Some asters will be formed from these centrioles in the animal cells, asters. And even spindle apparatus also will be formed. Here you can see that spindle fibers will be formed from that centrioles, and that centrioles will be forming the asters. Asters are star shaped structures. So, that will also be formed in the end of the prophase, and it will be forming the spindle apparatus. That is the end of the prophase, spindle apparatus. I hope all of you understood the prophase. Any doubts are there in the prophase? Asters, star shaped structures, formed from centrioles. Okay, they will be looking like this with two centrioles which are perpendicular, and they will be radiating out some kind of microtubules like this. So, it looks like as star shaped structure. So, this is one aster. Like this, two will be there at each pole. And from here only some threads will be coming like this. These are called spindle fibers. The pink ones are spindle fibers and these white ones are centrioles. Okay. So, from the asters only spindle fibers are also formed. What is the function of asters? To hold the spindle fibers at the poles. They will be present at the poles, and they will be holding the spindle fibers necessary for cell division. Clear? Yeah, spindle fibers are thread-like structures which are made up of microtubules. They are necessary for holding the chromosomes and distribution of chromosomes during cell division. Now let us see the metaphase. In metaphase, the spindle fibers will come and they will be attached to the kinetochores of the chromosomes. I will show you here clearly. Each chromosome will be looking like this with two chromatids. Okay. On either side, some small disc-shaped structures will be there like this. These are called kinetochores. On either side of the centromere, two disc-shaped structures are there. They are called as kinetochores. So, from the poles, the spindle fibers will come and they attach to the kinetochores from the asters. These are asters, these are spindle fibers. And you know, these white ones are chromosomes. Okay. So, from both the poles, the spindle fibers will come and they will be attaching to the chromosomes in this way. So many will be coming like this and they will be arranging together now to form the spindle apparatus. This total structure now, it is actually called as spindle apparatus. That initiation will begin in the metaphase only. Okay. First, kinetochores will be there on either side of the centromere. For that, the spindle fibers will come from opposite poles and they will be attached. That is the first event, attachment of spindle fibers to the kinetochores of the chromosomes. The next event, okay, all the spindle apparatus is already formed in the prophase only, attachment only will happen here. And after that, all these chromosomes, because of the spindle fibers are like threads, like kite pulling and all, like that, the spindle fibers will be pulling and pushing the chromosomes from both the poles, so that all the chromosomes will be arranged on the center of the cell to form the metaphase plate. You can see here both the chromosomes are present in the center. So here what happens is, in the metaphase, both the chromosomes will be placed in the center of the cell and they are attached to the both the poles like this. So we have here two kinetochores for each chromosome and they are attached to that poles by this. Here we have the asters. So in a cell, as the nucleolus and nuclear membrane, everything is not there, the chromosomes are present in the cytoplasm now. So because of the contraction, okay, and relaxation of the spindle fibers, all the chromosomes which are already attached to the spindle fibers, they are brought to the center of the cell to form the metaphase plate or equatorial plate. It is called as metaphase plate, or it is also called equatorial plate. So first event, attachment of the spindle fibers to the kinetochores of the chromosomes in the metaphase. Second event, the spindle fibers will be contracting and relaxing so that all the chromosomes will be brought to the center, exactly to the center of the cell or equator of the cell to form the metaphase plate or equatorial plate. This is the second event. So two events will happen, attachment of spindle fibers to kinetochores. Second one, metaphase plate formation, so that all the chromosomes are brought to the center of this cell or equatorial plate. Formation of this is very essential for the division of the chromosomes or cell division to be completed. Okay, this is the metaphase. Any doubts are there in the metaphase? Any doubts? So we have completed prophase and metaphase. Now we'll proceed to the anaphase. Okay, anaphase. All of you can see here, in anaphase, this type of chromosome exactly it is dividing in the middle like this. Here only two chromosomes are there, 1 and 2 chromosomes, red and blue color one. Now red one is splitting up into 2 chromosomes, daughter chromosomes, and blue one is also splitting up into 2 chromosomes. So in the cell in anaphase at this stage, we have 4 chromosomes. Let us see how that happens. This type of chromosome which was there, okay, that is already attached to the spindle fibers like this from both the poles. Remember, from both the poles the spindle fibers will be attached to these chromosomes. Now this one will exactly split in the middle. It will split like this. When it splits, no, the formed structures will be having only one chromatid each. So here 1 chromosome, 2 chromosomes are there. Here we have 1, 2, 3, 4. Now these are called daughter chromosomes. These are formed by the splitting of centromere. Centromere splits. And it will form daughter chromosomes. And these daughter chromosomes, okay, will migrate when they are moving. Okay, they will be moving towards the opposite poles. First, each chromosome exactly it will split in the center at the centromere region longitudinally, and it will divide into daughter chromosomes. One chromosome will become 2 daughter chromosomes. Now, parental chromosome is having 2 chromatids, whereas daughter chromosome is having only 1 chromatid. And that type of daughter chromosomes, after the formation, they will try to move towards the respective poles. Because already they are connected to the spindle fibers, they will try to move towards the opposite poles, the daughter chromosomes. So when the daughter chromosomes are moving to the opposite poles, no, these daughter chromosomes first, in this, centromere will be going first, then followed by arms. First is centromere, then second is arms, trailing behind. Like this, it will be pulled towards the poles. When they are migrating towards the opposite poles, first centromere will be going and followed by the arms behind. So while going, they will take different shapes like V shape, L shape, J shape, I shape, like that. Depending on the position of the centromere, you will get V shape, L shape, J and I. So in the anaphase, the changes what are happening are, the first change, centromere splits and it forms the daughter chromosomes. And then second point, the daughter chromosomes migrate. Daughter chromosomes migrate towards the opposite poles. Or move towards the opposite poles. Okay. So when they are going, they will be taking different shapes like V, L, J, I. So in this anaphase, daughter chromosomes are already formed and they have migrated towards the opposite poles. That is the end of the anaphase. Now the cell will enter into the last stage of the karyokinesis, that is telophase. Now let us see the telophase. All of you can see here the telophase. Telophase is having 2 daughter nuclei. Whatever the chromosomes that have reached here, they will be decondensing into chromatin and it will be dividing one nucleus into two daughter nuclei. So in the telophase, actually the events of prophase will be reversed. And by the end of the telophase, before we have started with one nucleus, okay, it will become two nuclei. That is the end of the telophase. So first reverse events of prophase will happen in the telophase. You know, in prophase I have told you that chromatin will be condensing into chromosomes. Now the event of telophase will be chromosomes will decondense into chromatin. Chromosomes are there like this, now they decondense into chromatin. They will be elongated and they will become the chromatin. Next event, okay, the Golgi complex, ER, nucleolus, nuclear envelope, all of them will be reappearing. They reform again. And then all the nuclear envelope, if it is formed around that nuclear material, obviously nuclei will be formed with the nuclear envelope. Okay. So two daughter nuclei will be formed by the end of the telophase. All the events of the telophase are exactly opposite to the prophase. In the telophase, events are opposite to prophase. The first one, whatever the single chromatid it has come towards the ends, this will become chromatin like this. So this is called decondensation of chromosomes to chromatin. That is the first event. So whatever the short rod shaped chromosomes are there, the daughter chromosomes, they will be enlarging or enhancing their size because of the decondensation process to become the chromatin. Next one, nuclear envelope. Nucleolus, Golgi, ER, reappear or reform. That means around this, here we will have one nucleolus, then around that nuclear envelope is formed. This is the nuclear envelope. This is nucleolus. These are the nuclear changes. In the cytoplasm, if you see, Golgi and ER are reformed because in the prophase they disappear. All these four disappear. In the telophase, all of them will reappear. Okay. So by the end of the telophase, one nucleus will become two daughter nuclei. Have all understood, ma? All of you see here. In the prophase, you can see asters are there. Asters are star-shaped structures which are formed by two centrioles and the spindle fibers which are coming from that, which will radiate. Microtubules will be radiating out of that to form that asters. From that asters only spindle fibers are formed. So that asters will be migrating towards the opposite poles in the cytoplasm. But in the nucleus, what changes will be happening? Nuclear membrane, nucleolus, Golgi, ER, they all disappear. And this chromatin will be changed into chromosomes. So by the end of the prophase, you can see that long chromatin will be changed into chromosomes. Then in the late prophase, you can see that asters would have migrated towards the opposite poles and they have formed the spindle fibers. Now it is looking like a spindle apparatus. This total structure is called as spindle apparatus. Now the cell will be entering into the metaphase where the spindle fibers will come from poles and they will attach to the chromosomes kinetochores, and all the chromosomes are brought to the center of the cell to form the metaphase plate or equatorial plate. Now in anaphase, the centromere splits in the each chromosome and it results in the formation of two chromatids separately, and each separated chromatid will be there, and that separated chromatids, they try to move towards the opposite poles with centromere first and followed by behind arms. Then in telophase, you can see here all the chromosomes which are reached the poles, they are becoming the uncoiling, they are undergoing uncoiling process and they are becoming the chromatin. After becoming the chromatin, nucleolus will appear, nuclear envelope will appear, Golgi and ER also appears. So, prophase events and telophase events are exactly opposite to one another. So, one nucleus in the prophase will become by the end of the telophase 2 daughter nuclei. This is the karyokinesis. Okay, the next one, cytokinesis. Cytokinesis means cytoplasmic division. Already I have written there, cytoplasmic division. In the plant cells, cytokinesis occurs by cell plate method. It occurs by cell plate method. In animal cells, it occurs by furrow method. In the plant cells, how it will happen is, here already daughter nuclei are formed, 2 daughter nuclei are formed already with the nucleolus, nuclear envelope, 2 daughter nuclei. These 2 are daughter nuclei. So, here we have the spindle fibers are already present like this. So, they will be arranged in the center, particularly Golgi vesicles will accumulate. This is Golgi. Golgi vesicles will accumulate in the center of the cells first, and then this Golgi vesicles will be growing towards the ends of this cell. Towards the ends of this cell, it will be growing, that Golgi vesicles, they will be going like that. And now it will become a phragmoplast. A barrel shaped structure is formed by that. That is called as phragmoplast. It will be growing from the center towards the side. So, this type of growth is called centrifugal growth. Centrifugal growth will be happening. Golgi vesicles will be joined. First, they will be placed in the center and they will be growing towards the side, which results in the formation of a barrel shaped structure that is called as phragmoplast. This will be further growing centrifugally, and now this will form the cell plate. Exactly in the middle, the cell plate will be formed like this. And here all cell wall materials will be deposited, and now the cell plate will be turned into the cell wall. So, now it is dividing one plant cell into 2 daughter plant cells. So, this is how the cytokinesis will happen in the plant cells. In animal cells, how it happens is, 2 daughter nuclei are already there with chromatin and nucleolus. Okay. Now, the spindle fibers are already there here. The cell will constrict in the center. This is the plasma membrane. It invaginates. It will be tucked in. And this tucked in plasma membrane, it will be growing towards the center. So, it will be growing slowly like this from both the ends. And after that, it will be fusing. Both the walls will be fused. The plasma membrane will fuse. And now it becomes a single one like this. So that it will be meeting like this. And it will divide the animal cell into 2 daughter animal cells. So, here it is beginning in the sides it begins, and it is proceeding to the center. This type of growth is called as centripetal growth. And whatever the structure that is formed here due to the plasma membrane invagination, this is called as furrow. You can call it as cleavage furrow also. Furrow will be formed in the parent cell, which divides that parent cell into 2 daughter cells. Okay. So, in the plant cells, it will happen in this way by the cell plate method, like which will be growing in the centrifugal way. And in the animal cell, the cell membrane invaginates at the sides first, and it will be growing towards the center, and towards the center means centripetal growth will happen, and it divides that by the furrow formation into two daughter animal cells. Next one, significance of mitosis. Significance. Mitosis is important for growth and even reproduction. It is required for cell repair. Okay, replacement of cells. To maintain nucleocytoplasmic ratio. Nucleocytoplasmic ratio. Okay, for all these things, okay, mitosis is required. Growth and reproduction in unicellular organisms is occurring by mitosis. Only growth in multicellular organisms is caused by mitosis. Cell repair, any damaged cells are there that will be repaired by the mitosis. Then replacement of old cells, dead cells, that is also done by the mitosis. Then maintenance of nucleocytoplasmic ratio. When a cell is growing, it will have more amount of nucleoplasm and cytoplasm. So, in order to maintain that nucleocytoplasmic ratio also, mitosis will happen. That is the significance of mitosis. I hope all of you understood. Any doubts are there? Occurs in both diploid and haploid cells. Yes, correct. But yeah, in the plant cells, it occurs mainly in the diploid cells. If any haploid cell is there, it can undergo. In plants, diploid cells. In animals, we have honeybee, which is haploid. So, it can happen in both diploid and haploid cells, majorly in diploid cells. Diploid means which are having two sets of chromosomes. No doubt? Okay, all of you see here. Try to understand the topic karyokinesis, cytokinesis. In karyokinesis, prophase, metaphase, anaphase and telophase are there. Cytokinesis, in plant cells it occurs by cell plate method, in animal cells it occurs by furrow formation. Okay, read this and come tomorrow to answer the questions. I hope all of you understood. Okay, bye. Thank you, Ma. See you tomorrow.
Hello, good morning students. So previous class we discussed about human skeleton. We discussed about axial appendicular. Axial skeleton has 80 bones, appendicular has 120 bones plus 6 girdles. So therefore 80 plus 126, that will be 206, right? So today we'll be mostly focusing on most relevant chapter for a biologist, most relevant chapter for medicinal field, pharmaceutical fields, and most relevant chapter for you also. This was health and disease. Okay, now how to define health? Health means a person, male or female, they must be physically healthy, psychologically, mentally healthy, and they should also maintain a proper communication with the society, and their intention should be normal. A physical, emotional, socio-communicating creature of human origin could be referred as healthy individual. There are some people who are physically healthy but not mentally healthy, like terrorists, Naxalites. They're physically fit but not psychologically. And there are some people who are psychologically very healthy but not physically, like handicapped people. They were psychologically very strong but not healthy enough, isn't it? And even these killers, murderers, they are both physically fit, mentally fit, but their communication skills were not good enough, so that they become anti-social elements, criminals, like that. Now, health could be defined as physically fit, mentally fit, and socio-economically, societally also
a stable individual. Guys, when you are healthy, when all your body systems are very, very active and acting or behaving to their maximum fullest, when you're healthy, all your body systems, digestive, respiratory, cardiac, excretory, muscular, bone, endocrine, nervous, if everything was normal in your body, then you will feel very easy to do any task given to you. Ease. So when you're physically fit, when you're healthy, each and every task assigned to you will become very easy. You'll show ease in doing the task, isn't it? You respire easily, you bend easily, run easily, digest easily, become healthy. That was health, right? But if any one of your systems, because of a pathogen or pollutant or enzyme or hormone, any metal ion, metal atoms, disturbs your system, any pathogen, pollutant, metal ion, hormone or enzyme, if it disturbs your health temporarily or permanently, then this ease to do any task will be disturbed, right? If any pathogen, pollutant, hormone, enzyme, metal ion disturbs any one of your system temporarily or permanently, you are not healthy anymore. You have lost ease. Losing ease, you have got disturbance in ease to do that task. What do you call loss of ease? Opposite of ease, dis-ease. So ease is healthy, dis-ease is not healthy. Dis-ease is a simple English term. It has no Greek or Latin origins. Simple English. Is, dis-ease. Like respect, disrespect. Appear, disappear. Ease, dis-ease. So when everything was in right place, if you're healthy, everything will become easy for you. Ease. But even if one system got disturbed, you will lose that ease temporarily or permanently. So losing ease was referred as dis-ease. Dis means opposite. Opposite of ease was dis-ease. Okay. Now the thing was that how a disease may happen. Does this disease happen to you? Does it originate in your body? Does it spread to other people? The disease you got this from other person or animal or a plant or a fungus? We do not know exactly. So our doctors and physiologists and pathologists study of diseases and their possible treatments was called pathology, and a person who did that was referred as a pathologist. Okay, so disease. So why our systems are not functioning at their maximum capacity? Because you got disease. And if it is a disease in your body, discomfort in your body, what was the main reason? Do you get this disease because of pathogen, or is it because of any hormone, pollutant, enzyme or metal ion? That could be traced easily. Now So if a disease was caused by a pathogen, a pathogen might be virus, bacteria, protist, fungus, sometimes animal kingdom, elements also. If there is a pathogen involved in disturbance in the body systems, this pathogen want to spread to other people also. So it may start in you and you may become a spread device to other people also. Therefore, such diseases which could be spread are referred as communicable diseases, or synonymously we can call them as infectious diseases. It might be because of bacteria, fungi, protozoa, or animals like helminths, and very worst of them, almost incurable disease, were in viruses. All these pathogens can cause you communicable or infectious diseases. But if disease is not because of any pathogen, there is no culprit behind your disease, there is no involvement of pathogen, it might be because of a pollution, missing some hormone in your body, missing some enzyme in your body, you got disease from your parents genetically, or your cell cycle did not regulate properly, your metabolism was disturbed because of any other reason but not because of pathogen. Such kind of diseases may happen to you, but they may not spread to other person easily. If there is no pathogen involved, chances of spreading the disease to other person was zero. Such diseases were referred as non-communicable diseases. Or we may also call them as non-infectious diseases because there is no pathogen involved here. Non-infectious diseases. So broadly, communicable, non-communicable, infectious, non-infectious. So let me give some examples of these non-communicable diseases happening to us. It might be because of excess or deficiency of some hormones. It might be because of too much or too low concentration of some enzymes. And these are possibly genetical also. It might be because of pollutant. And sometimes your cells would not listen to you. Mitosis may fail to stop. It happens continuously, which leads to a dangerous condition to the entire human body, that was cancer. Right? So you can see that communicable diseases and non-communicable diseases. Yes, Down syndrome is genetic. Down syndrome is a genetical chromosomal disease. Cancer, Down syndrome is non-communicable. Cancer, non-communicable. Good. Like this, broadly, simple. Communicable, non-communicable. Guys, when we speak about communicable diseases which could be spread or infectious diseases, so there are many categories. I will let you know some commonly asked, some commonly dealt diseases. So we can broadly split them into at least 4 to 5 categories. Simple enough. So diseases caused by bacteria. Done. Diseases caused by fungi. Diseases caused by protozoan or protistans. You can write here protista or protozoa. Same time, diseases caused by helminth, platyhelminth and nematohelminths, tapeworms and roundworms. And the most dangerous diseases caused by viruses. These are mainly 5 categories which are actually acting as pathogens for us. So we'll see. I'll give you some examples. We'll discuss upon that. So very famous bacterial diseases they include. So typhoid fever caused by Salmonella typhi, which could be a very dangerous fever, and it can also infect your digestive system, and typhoid fevers could be diagnosed with the help of Vidal test. So very common, typhoid. Next, cholera. Even more dangerous disease, which is an epidemic in many countries. Once in 13th, 14th centuries, when vaccines were not yet invented, cholera was the main cause of death in most of the countries. And you can go for one more, leprosy. Leprosy and lung associated tuberculosis. And do not eat stored food outside fridge. If you want to store food, store it in a refrigerator, but do not store them outside, because there is easy chance of food poisoning. Food poisoning is called as botulism. That is food poisoning. Don't eat stale foods. Okay, again, diphtheria by Corynebacterium diphtheriae. Whooping cough, what do you call that as pertussis. And rare skin disorder, that will be anthrax. Guys, I'm giving you names of these diseases in the context of humans only. I'm not giving you any examples of plant diseases. No. Very commonly you see diseases in humans. See, typhoid, cholera, leprosy, tuberculosis, botulism, diphtheria, pertussis, anthrax. Now, it's up to you. Try some. Give me some names of diseases which are caused by bacteria. Bacterial diseases. Try it. Gonorrhea, very good. Yes, it's an STD. Sexually transmitted disease. Yes, we can write that also. TB. Yes, wrote it. Tuberculosis, TB. Pneumonia. Very good. You can write that. Respiratory infection, pneumonia. Okay, yeah, that's okay. Good. Common diseases which are caused by fungi. There was a black mold known as aspergillus. If this fungus gets into lungs, this will slowly take down the immune system, and a person may have to suffer at least for 6 months to 1 year. That rare fungal disease inside your lungs is called as aspergillosis. Okay, and sometimes you may have, if anything on your skin leaves wet, if there is wet region of your skin, that wet part may get infected by some fungi and it may form a ringworm, like epidermophyton, trichophyton, fungus were there. Yes, ringworm is allergy, correct. And even some fungi were also responsible for dandruff. Fungi living in your hair roots may kill your skin tissue, which fall off as a dandruff. Okay, commonly found fungal diseases. And coming to diseases caused by protozoans, mainly the most dangerous of all was that it is caused by Plasmodium. The disease was malaria. Malaria is a protozoan disease, and if you ever eat food which was prepared at an unhygienic location, food which is contaminated with Entamoeba histolytica, you will have loose motions and stomach abdominal pain. That disease was amoebiasis or amoebic dysentery. Yes, when amoebiasis was caused by protozoan. And there is a notorious pathogen. If it comes into your body through a fly, there is a fly, let's say fly. If that fly infects this pathogen, right now you're sleeping 6 to 8 hours in a day. But if this pathogen comes into your body, it will mess up your sleep cycles and you will start to sleep 18-20 hours a day. This was referred as African sleeping sickness. African sleeping sickness. And it is also called as trypanosomiasis because it is caused by Trypanosoma gambians. Trypanosomiasis. Good. And speaking about infections caused by helminthes. Helminths include both platyhelminths and aschelminths. This include teniasis first. Infection by Tinea solium, teniasis. Flatworms, platyhelminths. And infection caused by Ascaris lumbricoides, aschelminth, ascariasis. And a dangerous pathogen called as Eukaryotic bancrofti, which may cause filariasis. Filariasis is also known as elephantiasis. These are some examples of helminth diseases. Now, guys, challenge for you. Yep. Get me some at least 20 different names of viral diseases. Try this. 20 different types of viral diseases, you know. Yes, I'll count. Number 1. Very good. COVID SARS, we call that as. Again, HIV, human immunodeficiency. For HIV is the virus name, it causes AIDS. HIV is the name of virus, it's not disease. You usually call that as AIDS. Come on, fast, even more. Again, measles, viral. Mumps, viral. Chickenpox, viral. smallpox, viral, polio, happen into spinal cord, viral, rabies, viral, some more. Flu or influenza. It is caused by flu viruses and bacteria actually. So flu, you have bird flu, swine flu, common flu. Very good. Again, come. Even more. Yes, good, very good. Hepatitis. By hepatitis virus, hepatitis. Inflammation of your liver. One more. Happening to your reproductive system, herpes, STD, genital herpes. One more. Common cold, rhinovirus, common cold. Even more. HPV causes genital warts, human papillomavirus, STD. Very good. Dengue virus, dengue fever. One more. Ebola. One more. Nipah. Very good. Nipah. Nipah was the Nipah disease. Zika. Some more. Try this. Get as many as you can. Try this. Very good. Chikungunya. Gunya fever. Very nice. See, so among all these pathogens, who is the ruling? Who is the king of pathogens? Is it bacteria or viruses? Obviously, unanimously, the answer must be virus. Virus was the most dangerous pathogen in our human history because most of the viral diseases are almost incurable and they know mechanism how to bypass, how to hide from our immune system also. Viruses are obligatory parasite. They want to live inside your cells. Outside a cell, virus dies. Inside a cell, virus lives. So virus is neither living nor non-living at that moment. If virus is out of a cell, non-living. If virus came into cell, living. That's why even in 2 kingdom, 3 kingdom, 4 kingdom, 5 kingdom, 6 kingdom classification also, virus has got no place because it is neither living nor non-living thing. Okay, these are communicable diseases which we may discuss. Done? See this big list of diseases you may get. All in humans. Then the other list, what was that? List of non-communicable diseases which do not require any pathogen or non-infectious disease. They might be because of some hormone. Excess hormone is also dangerous. Deficient is also more dangerous. Might be because of hormones. Might be because of deficiency or excess of some vitamins or minerals. Vitamin deficiency diseases. Might be diseases because of some pollutants. Might be disease because of error in metabolism or cell cycles. And the last one, disease which you get from your parents, genetical. It's a big list. Now, examples. Let us go into examples. Easy. hormone deficiency or hormone excessive diseases. So which include dwarfism because of deficiency of pituitary growth hormone, cretinism due to deficiency of thyroxine in pregnant females, diabetes mellitus, sugar disease because of deficiency of insulin in your body, diabetes mellitus. Same time, Addison's disease due to deficiency of corticosteroids of your body, Cushing syndrome, excessiveness of steroids in your body. All these are some examples. That's it. And coming to vitamin deficiency disease, guys, if your food does not consist of enough of B1 vitamin, thiamine, you may develop a disease called as beriberi. If you do not have niacin or nicotinic acid in your food, you may get pellagra. If your food does not have B12, you may get anemia, pernicious anemia. If your food does not have vitamin C, sour vitamin in limes, C vitamin. If you do not have vitamin C, then you may develop a disease referred as, deficiency of vitamin C cause which disease? Scurvy, bleeding gums. Not having enough vitamin D, rickets, knock knees, bow legs. Not having vitamin A, night blindness, nyctalopia. So, like this. Okay, some examples. And what about diseases caused by pollutants? So, pollution-based diseases. It may include asbestosis, inhaling too much asbestos dust. Okay, plumbism, minamata, mercury disease. Minamata is because of ingestion of mercury in your food. If you eat too much, food has too much cadmium, you may get itai because of excess of cadmium. These are some examples. What about error in metabolism or cell cycle? The most common cause of death as of now, leading cause of death in human history as of now. If your cell cycle does not get regulated properly, you may develop cancer. What do you call study of cancers? Oncology. A very, very dangerous form of disease, and cancer as of now has no cure, no medicine, no vaccine. There are possible treatments which may reduce spread of cancer, but no drug has been completely cured cancer. Okay, and genetical, guys, these genetical errors are because of loss of a gene, gain of a gene does not acting properly, or gene performing some overaction. Sometimes it happens. Loss of a chromosome completely, or sometimes gain of a chromosome also. All these are confined to DNA chromosome chromatin. If defect happens because of your DNA, those might be transmitted to your children also. So probably genetic diseases and enzymatic diseases are almost same, because enzyme is a protein which should be made from a gene. If a defect is in gene, that was because of defective genes. Therefore, genetic diseases and enzymatic diseases are almost same at the point. So examples, give me some examples of genetic disorders. Try fast, guys, try them. Genetic diseases: sickle cell anemia, thalassemia, hemophilia. The people with hemophilia cannot clot their blood in time or may not clot at all. A person cannot see colors properly, color blindness. It's very not dangerous, but sometimes. One more. Phenylketonuria, albinism, myotonic dystrophy, muscle degenerative disorder. Chromosome errors include Down syndrome, Klinefelter syndrome, Turner syndrome, sterile females. Klinefelter was sterile male, Turner's was sterile female. Many more were there. So once you come here, we'll discuss some of them in detail. The main root cause behind these diseases, how it has happened, and what is the consequence of the disease, and is there any way to treat it or prevent it. Okay? Good now, as of now? Done? Yes, there are some more diseases in which your own immune system will attack your own cells. Those are referred as autoimmune diseases like rheumatoid arthritis, Hashimoto's, myasthenia gravis, systemic lupus erythematosus. There are a few more. Okay? Yes, lupus is an autoimmune disease, systemic lupus erythematosus. Okay, that is done. So till now we just conclude here. Let us learn them in detail when you come here. Okay, we'll meet tomorrow. Thank you.